Good afternoon, dear students. My name is Bulat Aydana. I am a teacher of the Molecular Biology and Medical Genetics Department. So today's topic is genomic mutations. Here is the content of the lecture. Introduction, definition of the genomic mutations, types of the genomic mutations, causes, diseases arising from the genomic mutations, and conclusion. Mutation. To discuss about the genomic mutation, we will start with the definition of the mutation. Mutation is a natural, spontaneous, or artificially induced change in the genetic material. So this change is heritable. Mutation term is introduced by the Dutch physiologist Hugo de Vries in 1901. Also, he proposed his mutation theory based on this term. On the scheme, you can see classification of the mutation based on the level of the change in the genetic material. Gene mutation, chromosomal mutation, genomic mutation, and cytoplasmic mutation. Gene mutation is a change in the DNA sequence. Chromosomal aberrations, it's a change in the chromosomal structure. Genomic mutation is a change in the number of the chromosomes, and cytoplasmic, it's a change in the DNA of the mitochondria and chloroplasts. Today we will focus on genomic mutations, which result in the changes in the number of the chromosomes. There are two types of the genomic mutations, polyploidy and aneuploidy. Polyploidy is an increase in the number of the entire chromosome sets. For example, we say that human chromosomes are diploid because each chromosome present in two copies. If there are 3 copies of them, it will be triploid. 3n means triploid, also considered as a polyploidy. Tetraploid, 4n, 4 copies of each chromosome. 5n means pentaploid, and 6n means hexaploid. And aneuploidy, it's a different group of these genomic mutations, means change in the number of individual chromosomes. Here are the types of the polyploidy. They are further divided into autopolyploidy and allopolyploidy. Autopolyploidy arises through chromosome duplication within a single species without hybridization. It also may arise when a diploid gamete will be fertilized with a haploid gamete, and this produces triploid organism. Example is cultivated bananas, and common in plants such as grasses, crops, and trees. Polyploidy is uncommon for the human, it's common for the plants, trees, and grasses only. Allopolyploidy arises through hybridization between two different species, followed by chromosome doubling. Example is a cross between cabbage and radish produces fertile plants with the combined chromosome sets. So here you can see the mechanism of the polyploidy. Under the influence of the radiation or temperature, the homologous chromosomes of all pairs are not separated during the meiosis, and the abnormal gamete, as you can see on the second parent, diploid gamete was involved in the fertilization, and in the offspring is triploid. So aneuploidy, as we said before, it's a change in the number of the individual chromosomes. You see that 2n set remains the same. There are 4 different types: nullisomy, monosomy, trisomy, and tetrasomy. Nullisomy formula is 2n minus 2, means there is a loss of the both homologous chromosomes. Monosomy, 2n minus 1, means loss of the one individual chromosome. Trisomy, 2n plus 1, means there is an extra 1 chromosome, gain of 1 chromosome. And tetrasomy, it's a gain of the 2 extra chromosomes. So the cause of the aneuploidies is non-disjunction. Non-disjunction, it's a failure of the chromosomes to separate properly during the cell division, mainly in the anaphase. So here are 2 possible stages where the non-disjunction can occur. It's the anaphase 1 and anaphase 2. In the anaphase 1, homologous chromosomes should separate and move to the separate poles. As you can see from the left side, in the first division, homologous chromosomes move to the same pole and formed in the one cell, while the second cell is empty. After the second division, there are 4 abnormal gametes are formed, and when it's fertilized with a normal haploid complete gamete, they produce trisomy and monosomy organisms. Then this non-disjunction may occur in the anaphase 2 also. in the second division of meiosis. In this phase, sister chromates should separate into the different poles, but sister chromatids remain in one cell. So when these abnormal gametes fertilized with the haploid normal gamete, they also will produce trisomy and monosomy. As you see here, 4 organisms are produced, 2 are normal disomic and 2 of them are abnormal, trisomic and monosomic. So what is chromosome? Chromosome, it's a structure located in the cell nucleus that carries genetic information and ensures the transmission of the genetic information from the one generation to the next. It consists of DNA and proteins. So what is karyotype? It's a complete set of chromosomes of an organism, including number, size, shape, and structure. Chromosomes can be autosomes and sex chromosomes. Normal male and female karyotype you can see in this gray box. Male karyotype is 46,XY and the female karyotype is 46XX. Autosomes, there are 22 pairs of the autosomes. These are the same in both males and females. They are numbered consecutively from 1 to 22. Sex chromosomes are the chromosomes that contain genes responsible for determining the biological sex of an organism. So females produce only X-bearing gametes, so they are homogametic, while the males produce X-bearing and Y-bearing gametes, so they are heterogametic. According to these types of the chromosomes, aneuploidies are divided into 2 types, autosomal aneuploidies and sex chromosomes aneuploidies. Autosomal aneuploidies means number of the autosomes change. Here you can see the examples: Down syndrome, Edwards syndrome, and Patau syndrome, and also the karyotypes for these syndromes. Trisomy 21 or Down syndrome karyotype is 47XY plus 21. This syndrome is described with the upward slanting eyes, flat nose and face, thin upper lip, opened mouth with a protruding tongue because they have macroglossia, large tongue, small ears, excess nuchal skin, means excess skin on the neck, short neck, brachycephaly. Also, here is the realistic picture of this syndrome patient. Here is the karyotype described, Down syndrome and patients. They will have also problems with the cardiovascular system and aortal dissection and dilation. Next syndrome, autosomal aneuploidy, is trisomy 18, Edwards syndrome. Karyotype is 47XX, 47XY plus 18. This patient has unusually small head, and the back of their head is prominent, ears malformation, very small jaw and mouth, micrognathia, hands are clenched into fists and fingers are overlapped, and club feet or rocker bottom feet. Also here in karyotype of this syndrome, you can see underlined with red 18 trisomy, 1 extra chromosome. These patients usually die within the several weeks of their life because of the pneumonia and infections. Trisomy 13, it's a Patau syndrome. Karyotype is 47XX, 47XY plus 13. Main symptoms are cleft lip and palate, small head, small eyes, microphthalmia, ear malformations, extra fingers, polydactyly, heart defects, spinal cord abnormalities, and kidney cysts. Average survival is fewer than 10 days. Here represented sex chromosome aneuploidies, Turner syndrome, Klinefelter syndrome, XXX, Jacob syndrome, or Y syndrome. They are not viable because there is no X chromosome that is responsible for the normal growth and development. Monosomy X, it's a Turner syndrome. Karyotype is 45XO. This patient will have short stature, webbed neck, narrow hips, poorly developed breasts, immature sexual organs, infertility, subnormal intelligence, lymphedema of hands and legs, swelling of their hands and legs, and according to the karyotype, there is no barred bodies. Klinefelter syndrome karyotype is 47XXY or 48XXXY. Main symptoms are they are taller than the average human, underdeveloped testes they will have, enlarged breasts, development of the breasts in the males we call gynecomastia, little hair on their body, infertility due to abnormal seminiferous tubules, mental retardation, small testes, obesity, weak bones, and many female characteristics. And they have at least one bar body. Triple X syndrome karyotype is 47XXX. They are above of the average height. Delayed speech development they will have, mild intellectual disabilities, menstrual irregularities, and premature or ovarian failure, but physically often they are normal. So here are the factors contributing to the genomic mutations. These are the maternal age over the 35 or paternal age over the 40, recurrent miscarriages, and menstrual disorders. So, conclusion. Chromosomal disorders are one of the most important issues of the modern medicine. In recent years, the frequency of the detection has been increasing, mainly due to improvements of the prenatal diagnostic methods. And among chromosomal abnormalities, Down syndrome is the most common. Providing children with these conditions with early medical, physiological, psychological, and social support can improve their quality of life, facilitate their integration into the society. Therefore, it's important to enhance genetic screening programs, increase public awareness of genetics, and strengthen preventive measures. Thank you for your attention.